Hello people, welcome to my channel Biology Updates. If you are new to my channel, please subscribe to my channel, and if you have visited again, then welcome back to my channel. Today we would begin with another fresh topic, and that is linkage. Linkage was described first by Morgan, and we would also we would discuss about X-linked diseases, autosomal and recessive X-linked diseases. So I have previously described about autosomal diseases and autosomal pedigree. If you want to look up to that video, you can go back to my channel and look for that video. I'll also pin it down for your easy reference. So let's begin with today's topic, X-linkage. X-linkage describes the genes, particularly on the X chromosomes, means the chromosomes, the gene set responsible for the sex of an individual. In many animals, at least one of the individual is showing heterogamity. For example, in birds, female is heterogamity, and in humans, male is heterogamity, which means that, what do you mean by heterogamity, first of all? One individual is having different sets of gamete. For example, in case of humans, Drosophila, we find male heterogamity. The female gamete, if you see, would be X, and the male gamete would be X and Y. So here, female is homogamity, and the male heterogamity is responsible for determining the gender of the progeny. Therefore, heterogamity is important. But in case of birds, we find female heterogamity, and we usually describe the female gametes as Z and W. In roundworm and insects, the female gamete would be XX and the male gamete would be X0. This is the case of roundworms and insects. They are also heterogamous. So here, the female is responsible for determining the gender of the progeny. But in humans, Drosophila, insects, the male is responsible for determining the gender of the progeny. As described before, autosomes are genes that are present, or that are genes that describe the other body functions. In our body, we have two types of genes. Or we have 23 pair of genes, and among which 22 pairs are autosomes, genes that do not determine gender of an individual. They are genes which are responsible for other functions like protein formation and different sort of signaling and structures, and immune system genes responsible for formation of WPCs, macrophage, and different kind of immune cells. But there is one pair of chromosome gene which is responsible only for sex influenced or sex limited, and which determines the sex of an individual. That is called as X chromosome. Only one pair of X chromosome. So earlier we only had idea of autosomes. We did not discover X chromosome. So after linkage was discovered by Morgan, the inheritance pattern because of X linkage will also change and shift. First case of X linkage was discovered or documented by Thomas H. Morgan, in the year 1910. He was experimenting with Drosophila, and he found mutation in white eyes, and that led to his understanding of X linkage. So Morgan considered two crosses in Drosophila, a white eye of mutation in the white eye of Drosophila, if we see in first cross, the female is red-eyed, and in the second cross, the female is white-eyed. And in the first cross, the male is white eye, and in the second cross, the male is red eye. So we have exchanged the characters, and what we found is the F1 and F2 generations were very different. Till now, the crosses that we have considered, we have seen that irrespective of the gender or the characteristic traits that we take, the F1 and F2 generation is unchanged. Even if we exchange the traits, the results of F1 and F2 generation would remain same. But now, in X linkage, Morgan found that the results were a bit different. In F1 generation, when the female was red-eyed, it did not produce any progeny with white eye. But in F2 generation, when the female is white-eyed, it produced half of the progeny males white-eyed. So next thing to consider was that in F2 generation, 1/4 was white, 1/4 males were white, and no white red. But in F2 generation of cross B, if you see, all the possibilities were visible. Half of the 1/4 of the population were red-eyed female, 1/4 were white-eyed female, 1/4 were red-eyed male, 1/4 were white-eyed male. So that led to conclusion that this particular trait type is present in the X chromosome and not in the autosome. And Mendel also hypothesized that the recessive allele for white eye is found on the X chromosome, but its corresponding locus is absent from the Y chromosome. Now, if you anyway see the structure of X and Y chromosome clearly, the X chromosome is a little bit bigger and the Y chromosome is a little bit smaller. Since the Y chromosome lacks homology with almost all the genes of X chromosomes, these alleles present on the X chromosome of the males will be directly expressed in the phenotype. Technically, males cannot be homozygous or heterozygous for X-linked genes. They cannot be carrier. They are therefore mostly diseased. If there is some kind of problem in X linkage, X chromosome in males, they usually they show the disease, and there is nothing called as recessive gene there. Therefore, the individual is called hemizygous. Hemizygous, why? Because they have two different type of gametes. When there is a male heterogamety, the males are hemizygous, and when there is female heterogamety, the females are hemigamous. In case of humans, if I consider, males are hemigamous because they have X and Y, two kind of gametes which are responsible for sex of the gender. Morgan and his students, Kelvin Bridge, provided the basis of chromosomal theory of inheritance. They provided solid evidence that genes are transmitted on specific chromosomes and they are present on specific chromosomes. And also, not the entire genes are transmitted from parents to offspring, but there is a specific part of genes or just specific type of genes that are being transmitted or inherited from parents to offsprings. The next thing that we're going to study is X linkage. In humans. Same as autosomal linkage, in human beings also we have recessive and dominant type. So first we would study about X linkage, X-linked dominant pedigrees. What are the characteristic traits of X-linked dominant pedigree? So traits are quite common. You would commonly find it because it's a dominant disease type. So you would find it in every generation and it is quite common. Second point, not skipping generations. This characteristic trait of autosome dominant pedigrees is it does not skip generation. It would be visible in every generation. The only important factor here is that multiple kids should be produced so that every possibility is possible. Next is affected fathers pass to all of their daughters. So sons are not affected, but affected fathers pass the disease to daughters. From father to daughter, the disease gets transferred. So this was all about X-linked dominant pedigree. Now, X-linked recessive pedigree. So in X-linked recessive pedigree, males are more affected and always mother to son, never from father to son. So males are affected because they have a carrier mother or affected mother, because of which males are more affected and females become carrier, and traits would skip generations because it's a recessive pedigree. So obviously, now we would look up to two pedigrees. We would understand how do you solve those X-linked pedigrees. Okay, so this is one pedigree that we would consider. Things to notice here is that the mother is affected in F1 generation, and in F2 generation 2 sons are affected, and in F3 generation only one son is affected, but none of the daughters are affected. So this can be X-linked recessive pedigree because none of the daughters are affected, and the trait is passing from mother to son. So let us solve. Male has two type of sex chromosomes, X and Y. So I am denoting X as like, this is a healthy male, so it would have a dominant X chromosome, and this is Y chromosome. Okay, the Y chromosome is normally shorter, but I'm denoting it a little longer than X1. So this is affected female, so she would be homozygous recessive, and she will have X and X. So 2X, therefore both are recessive, and she's healthy. So now she has two sons and no, two sons and two daughters. Both the sons are affected. So son will have one Y chromosome, and X chromosome is recessive type, therefore the sons are diseased. Now females would be carrier. Females would be carrier, otherwise they have no option. The father is normal, but the mother is homozygous recessive allele type, so females would be carrier. There is a higher possibility that females, when they reproduce, they would give birth to affected males. She's also normal female. Now this female got married to normal male, so again Y chromosome. But this is affected. One of the sons are affected, the rest are normal. This is also normal female, carrier but normal. And again, this is affected individual. This will have recessive allele. They will all be normal. So that's how the pedigree would look. This one homozygous recessive X-linked pedigree. This is classic example of color blindness. Insensitivity to green light and red light. That's what happens. So this is one pedigree. Second pedigree. If we see here, in first generation, the female is affected, and in the second generation, male and female both are affected. Here, if you see the second generation, the father is affected, the male is affected, and the disease is being transmitted to only to females, daughters, not the sons. And here, both are affected. Still, the daughters are affected here, but not the sons. As both are affected, and first thing, daughters and sons both are affected. Second thing to notice is that in this generation, in this pedigree, in this portion, the father is affected and then sons are not affected. The disease is being transmitted from father to daughters. And so I feel this pedigree would be dominant, but let us solve it. Pedigree solving is all about hidden trial method, so you have to use multiple approaches. But okay, so the male is XY, so one is X, this is Y, and this is, I'm considering it as dominant trait, so this would be A. Recessive would be normal, and single gene, single dominant allele can cause the disease. So this is, I'm marking it as heterozygous dominant allele for simplicity. And next is, this is again the diseased one. This is normal. AA. This female is normal. This male is also normal. This female is diseased. So again, one X from mother, another normal X from father. This is diseased son. So one Y is normal. X linked, so Y, it has nothing to do with Y chromosome. X chromosome is affected here. This X chromosome is being transmitted from mother to son, and therefore the son is having this disease. He got married to X. He got married to affected individuals. So yes, here also she is affected. We can see till now that this pedigree is being solved. We are able to solve it by dominant trick. This is ANA, and then again she got married to normal person. Writing it here. This is again affected son. Affected son is one affected, and this is Y. Affected daughter, normal daughter. Affected daughter. And normal son. Normal son is the Y chromosomes. This is normal son, so this will be all normal. Both the sons are normal. This is also normal son, normal male, but they have, she's affected, these both are affected. Normal son. Affected son will have, again last, one dominant allele from mother, which is causing the trait, causing the disease, in one Y from father. So we can solve this pedigree by using dominant trait. So let me write it. X linked dominant trait. Genes responsible for it is AA or AA. There are two possibilities. But if you see, if I mark it as AA, if this gene would have, you can see here that only few progenies are affected. Not all of the progenies in each and every generation is affected. So it is AA gene which is causing the trait. Diseased gene. Heterozygous. So there can be 2-3 type of questions. Is it heterozygous? What can be the gene? What is the nature of the pedigree? This is example of fragile X syndrome. There are other examples of X-linked traits like hemophilia, Hunter syndrome, etc. So this was all about pedigree solving. If you like this video and if you like my content, then please like, share and subscribe to my channel Biology Updates, and I will meet you in the next video with new fresh topic. Thank you so much. You can also comment down and let me know your thoughts about the videos and the content that I make. Thank you so much again. Bye bye.